I'll just go through the complete revision of the cell biology. What are the complete syllabus? I will just cover it. Hope that I can cover within 1 hour, or it might be 2 hours. So from the starting part, what we have already known about the what is a cell, and it's a basic definition. You know about the cell is a kind of smallest and the basic element, and that with the structural and the functional value of the fundamentals of unit of life. And also along with that, you know about the 98% of the mass, the composed of cell of it will be like carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. These are kind of major element that constitute the cells. And the approximately 70% of water containing on that, and also the like cell can evolve that can able to sense the natural response or kind of mechanical response to the environment. Along with that, the filled with some concentrated aqua solution in the chemicals, in form of chemicals or of chemicals, and having ability to create the copies themselves. That is the kind of basic definition of the cell. And the cell size and shape, it might be varied, depends on the cell, which type of cells you have, you are studying of that, or that can change, depends on the size and shape. And okay, now for few questions, you should know about some of the landmark of that cell. So what are the landmarks? Kind of discovered cell by Robert Hooke, kind of 1665. And then one of the major inventory of about the discovery of nucleus by Robert Brown. And like Swan cell or Slyden and Swan, that is the proposed of the cell theory, like how cell is evolved from a natural like kind of chemical compound to how a cell formed like a living conditions, that kind of proposed hypothesis. And then the Camillo Golgi, the kind of Golgi apparatus inventory in the 1898. So these are kind of major discoveries of the cells. So and then the what are the basic theory already established or kind of proposed, or we can accept it, like of evolution of cell from 3.5 billion years ago, and along with the fast organism like chemoheterotrophs. Already I have discussed in previous video, like what is the autotrophs and what is the heterotrophs. And in heterotrophs, one is the chemoheterotrophs, like chemical molecule that can survive and coming to that organism system, and that can make some of these basic fundamental unit, or taking as a food element. So along with that, also we have studied some of the chloroplast related in the plant biology, and some cell have some genetic material that might be the DNA and RNA. So already the nucleic acid will be there. They are kind of basic reproductive storage capacity, or reproductive storage things that can keep their genetic data on the DNA. So in structure, you all know cell have some boundary. So depend on the cells, it might be prokaryotic and some eukaryotic. Their differences are there. And if you talk about the cytoplasm and nucleus will be there. So you talk about the cytoplasm, that will be some of these like tropoplasm, and some of the like hyaloplasm. So or you can just easily break through these things. There's some of the cell organelles are there, and some of these non-cell living inclusions are there. What are the non-cell living inclusions? First I will go through that. First of like different kind of carbohydrate, and some of these nitrogen oxides, and some of this fat and oil. So these are the things that present on the reservoir molecule, you can say reservoir molecule of a cell, along with some essential oils and glycosides or organic acids, some minerals, some crystals, along with for plant cell it might be some alkaloids, gum, latex, resin, and the tannin. So these are kind of different elements are there in the non-living cell in the inclusions. And you say about the living part, that can be that might be like mitochondria, Golgi, and lysosomes and ribosomes, plasmid. And for the prokaryotic cell, it might be the cilia, flagella, and along with some eukaryote, like some of the microbodies are there. So these are kind of constitutes, these are kind of things are present in the cell. So now the different types of theory have been proposed in the theoretical part, like some theory like on previous days, like I think like 18th century. So some type of theories are there, theories are evolved, and currently those theories, some of the theories are kind of baseless nowadays, but still they are how the science is evolving on this purpose. So one of the major important theory is like the exception to cell theory, and this is the kind of bacteria and green algae lack of nucleus, like some exceptions in the cell theory, and like their virus lack of the protoplasm, like that the cells are made of nucleic acids and proteins. So now I will go through the eukaryotes and prokaryotes. You all know prokaryote is a kind of bacterial cell. My cell size will be small, and cell wall is not made up some of cellulose, and their ribosome is like 70s, the 50s plus 30s, and these are kind of basic things. And I'll go through more details on the more difficult things. And eukaryotic cell, it's large components of the large compared to the prokaryotes, and it's like the ribosome will be 80s, like 60s plus 40s, and they are but different kinds of RNA polymerases will be there. rRNA, messenger RNA, mRNA, and also tRNA will be there, and monocytonic DNA are common in that section. So these are kind of basic difference between these prokaryotes and eukaryotes. Also, some major differences are also there. Now we talk about the membrane, because in cell signaling and all those aspects, the membranes are essential. So cells will have the membrane, and you all know about that proteins and lipids. They are the major component of that. Different types of proteins are there. One will be the transmembrane protein, or lipid anchor protein, or will be like uh, transmembrane, lipid anchor, and another will be like peripheral protein. So these are the proteins are present in the cellular membrane, and lipid bilayer is there along with some cholesterol uh, are there. These are kind of major constituents of the cell membrane of eukaryotic cell membrane. Also, some of the cells they have their cell wall in plant cell, along with some gram positive and gram negative. Both their cellular structures are quite bit different, but for signaling aspects, so everything has some different type of signaling, and those are mainly present in the cellular membrane. Signaling molecules, and they are attached with some receptors, and receptors are nothing but like a different kind of proteins that present in the cell surface. Let's talk about some of the membrane lipid, what are the present in that cell. Some of this like integral membrane lipid, some of this peripheral. If it is integral of this membrane, like anchored, and some of the transmembrane, like that can pass. You see, if it is called transmembrane, it means it's a bilayer. In that bilayer, and that can pass through the inner to the outer portion, that's a kind of pass, and it pass might be the single pass, and along with some of the multi pass. So single pass, that might be two type, in the type 1 and type 2. And if you talk about the anchor, it might be lipid anchor protein, or might be the GPI anchor protein. So these are kind of proteins present in the cell. Okay, let's talk about some of the cell transport and how cell transport happen. So transports might be the passive transport, some of this active transport. And how you talk about passive and active, it's active, it's a loss of energy or ATP it's needed. So kind of that is active transport, and passive, it's due to the diffusions. So that have there some you called about the simple diffusions, and you called about the facilitated diffusion. So in the simple diffusion, that is osmosis will be there. Osmosis, if you know about osmosis, that might be the hypertonic or hypotonic, or might be the isotonic solutions. And if you talk about the facility diffusion, then the things are coming. One will be the channel protein and another will be the carrier protein. So channel and carrier. And active transport, the exocytosis and endocytosis there. So in endocytosis there will be two types, like kind of phagocytosis and the pinocytosis. Phagocytosis is very kind of solid molecule that can engulfed by the cells by the phagocytosis mechanism. If you talk about the pinocytosis, kind of liquid molecule in coming to the inside to the cell, so that is a kind of pinocytosis. So these are the kind of transport across the plasma membrane. And we talk about the membrane constitution or membrane protein constitutes. Now you think about the cell wall. Already I have discussed okay, so some of the examples of these active transport and passive transport, they are active transports like sodium potassium pumps. So in that pumps, usually active transport, it's take the energy. And if you talk about the primary active transport and the secondary active transport, like they uses the primary active transport uses the ATP to transport all the species in the solute across the membrane, and they against their concentration gradient. And if you talk about the secondary active transport, this kind of transport molecule across the cell membrane utilize the energy in other form of ATPs like GTP, CTP, this kind of energy molecule. These are kind of secondary active transports. Now you talk about the cell wall. This is kind of 10 minutes over. So now talk about the cell wall. Cell wall is defined as the non-living component because it's they are covering their outermost of the cell. Its compositions varies across the different organisms, and it's and also permeability of this nature, and permeable in nature depends on that. Also cell wall separates the interior components like of the cell in from the exterior environment. So cell wall normally absent in the animal cells. So now talk about the cell wall structure. I'll not go through the complete details. It's kind of revision. That's why the primary cell wall, some of the important element are the cellulose, hemicellulose, and pectin. So these are kind of they are constituents of their kind of primary components of the cell wall. It will be like cellulose, hemicellulose, and pectin. And in middle lamella, it's a layer, and there's a cements of this primary cell wall to adjoining plant cell together. It's a kind of fast form layer which is deposit at the time of the cytokinesis. The cytokinesis is a part of the cell division and after cell division procedure. So like two major ions are responsible. One is a kind of calcium ion and there is a magnesium ion. So now we talk about the secondary cell wall. So what are the components like pectin, cellulose, hemicellulose, and lignin? So the major difference is the primary and the secondary. The lignin is present in the secondary cell wall, and in primary cell wall, lignin is not present. So secondary cell wall found some of many plant cells and located between the primary cell wall and the plasma membrane. These cells start producing the secondary cell wall after their primary cell wall is completely or kind of stop expanding to outer cells. So these are kind of thing. And another important key aspect is their plasmodesmata. What is the plasmodesmata? Are the cytoplasmic channels that pass through the cell wall of the adjacent cell. Like it's a cell wall kind of non-living, but to transport this molecule from the one cell to another cell, so it's a kind of channel present the adjacent cell. So how they are pass their material to one cell to another cell. This is called the plasmodesmata. So now after that we'll talk about some of the primary cell wall and the secondary cell wall differences. So in that differences, you can the one of the major differences is kind of extension, component extension. And yeah, in secondary wall its extension is very low, or you can say generally absent. And another like a primary wall is just solely layered like one layer, and in secondary wall it will be like three layered component. So then talk about the nucleus. You will know about the genetic material stored in the nucleus, and they are They store their molecules for the cellular process and responsible for the giving cell shape, and all the machinery is present on the nucleus part. And then you can go through the cytoplasm. You know about all the cytoplasm is first discovered by the Strasberger in 1882. So it's a kind of gel like thing, and most of the in that gel the most of the things are water, kind of 80% of water, or you can say more than 80% of water are there. And then we talk about some organelles, because you know about the ER is one of the major components, like smooth ER and the rough ER. In the smooth ER part, like endoplasmic reticulum, then they don't contain the ribosome there, and also like RAP here they are heavy with the ribosome part. And another important protein like the riboporins. In riboporins, the in smooth part it's not there, but in the RAP here it is still there. So now the important mechanism, these are the components you study about that, and then your main focus will be on the major important like mitochondria. So why it is so important? Because they are energy mechanism, because this in exam part the mechanism section give the lot of questions. So, like cardiophyllin contains there are four fatty acids rather than two, help to make the inner membrane impermeable. So, so these are the important aspects. In the revision part, I will not go through the complete details on that. So, and ribosomal proteins, you already I have discussed about the subunits, but few proteins are like the R proteins and they are rRNAs. So, like in ribosome, like 70s, there 50s are subunits, are kind of subunit, and also RNAs are like 23rd A's plus 5s under 28 A's. So this is the RNAs and R proteins, over 31 proteins are there. I think sensor is not working properly. Okay. And along with 30 A's, their RNA is up to the 16 A's, and along there 21 proteins. These are kind of information. You just go through on it. Okay, and lysosome, and this is one of the key aspects. So, what are the major important things here? And the most of the questions are coming, and you have to remind is like their vesicular transport and the membrane fusions and the protein. So two type of pathway for vesicular transport. One is the anterogate pathway and the other is the retrograde pathway. So in anterogate pathway, their proteins packaged into the transport vesicle moved into the in forward manner, and in retrograde their packaged protein into transport vesicle move in the backward manner. So one is the forward manner and another is a backward manner, and along with some signals are there in retrograde. You all know about the KDEL signals or KKXX trival signal sequence are there in the carboxy terminal. So and for the anterogate pathway, it will be like ER Golgi intermediate component, and then vesicle Bardock wire and fused with the Golgi and the cis Golgi cisterna, and for the further shortening. So like proteins forward manner in anterogate, in retrograde in backward manner. So backward it needs the some of the sequence retrieval signal. Okay, so the concept is like that the Golgi apparatus that make some the cis trans Golgi network. In that network the secondary vesicle is formed, and then they move to the nucleus part for the secretion, move to the membrane part, and then it's kind of signal transduction occur, and then it's the regulated exercise occur. So these are kind of mechanism, and important is the signal sequences. They gram-positive bacterial membrane, bacterial wall, cell wall, and membrane composition, and gram-negative bacterial cell wall composition. So and cell membrane composition. These are the very important aspects for targeting antibiotics. So, and these are the part where the questions are coming often. And in plant cell wall, you talk about pectin, structure of pectin, and structure of hemicellulose, and how they bind. These are important, but for revision, you should taking care of that. And also, already I have discussed the plasmodesmata and their symplast and the apoplast. So in the in between the space of two cells, like 2 3 cells, the space is the inner space is the kind of apoplast. And some of these are continuous compartment from the different plant cell, it's called the symplast. Kind of continuous compartment is called the symplast. So also you can study about the tubular connections in the in the plasmodesmata. Okay, so protein trafficking, that's the important aspects. You should go through the complete details. Hope one day I will explain about then how the protein compartmentalization of the steps in the glycosylations and the subsequent modifications of the protein in the ear and the Golgi. So I'll go on other things. And in microtubules part and how they form their alpha beta, alpha beta, alpha beta, alpha beta, this kind of chains. And you know about that one alpha one beta would be like 8 nanometers. So then A tubule, doublet and triplet, and A tubule, B tubule and C tubule, how they attached with that the microtubule structure. And you can go through about details like cytoplasmic microtubules are simple tubules, and the singles microtubules are also built with their 13 protofilaments. Other things. Then you go through the human genome part. In human genome part, number of the exons, number of the introns, and classical human genes, like total length of the gene kind of 27 kilo base pairs, and some of this coding in region, some of the untranslated region, some of this like size of introns. So these are the human genomes of these actin filaments, and also the gene and non-coding gene. So some of these essential, like we have some gene families and pseudogenes are there. So that pseudogenes are not so important, but still you can just go through that. And then their eukaryotic cell cycle, these are the major part whether your understanding is good or not. So in your cell cycle part, and another key aspects is the regulator of the cell cycle, that is also part of it. So in next video, I'll explain about the some of the cell cycle and revision of the cell cycle, because it's a large zone. And another thing, yes, that's fine. In the cell cycle part, the major one is the cell cycle and the regulation of the cell cycle. In 2 3 books you can follow, but in revision section you can follow any books. Normally Lordis is there, and along with Carp cell biology books are there.